names are Diana,
Melissa,
and T.
The big question we will focus on is, how is DNA replicated for researchers to conduct studies in abundance?
The answer to the aforementioned question is relatively simple: PCR. So, what exactly happens during a PCR reaction? Let's find out!
PCR is used every day to diagnose diseases, identify bacteria and viruses, match criminals to crime scenes, and in many other ways. To perform a PCR reaction, you need DNA that has been extracted from cells. Start by moving your extracted DNA into a special PCR tube. PCR works by heating and cooling the solution repeatedly, so PCR tubes are designed for even heat distribution. Next, add primer 1 to the PCR tube. Primers are powerful tools in copying specific DNA sequences because they attach to the sites on the DNA strands that are at either ends of the segment you want to copy. Now, add primer 2, which will attach to the second site. Last, you will add the master mix, which consists of nucleotides and DNA polymerase. Now that the tube is filled with all these components, you will proceed to put the sample into the PCR thermocycler. Inside your PCR tube, cycle 1 has begun. The DNA heats up to 95 degrees Celsius in order to separate the DNA strand. This process is called denaturing. Then the temperature decreases to about 55 degrees Celsius, so the primers could bind to both ends of the sequence. This process is called annealing. Afterwards, the extension process occurs, causing the temperature to change to 72 degrees Celsius. In this step, DNA polymerase is activated, causing the primers to complete the sequence with nucleotides. After about 30 cycles of the same steps repeating, the target sequence is copied over billions of times.
PCR is short for polymerase chain reaction. The name itself is already self-explanatory. Remember, in order to further genetic research, labs must obtain a large quantity of DNA. Therefore, just one is not sufficient. PCR is a rather simple and inexpensive tool that you can use to focus in on a segment of DNA and copy it billions of times.
One question always leads one to inquire new questions.
Like, what do researchers do with the ample amount of DNA after the replication? What research process requires PCR?
but what we learned for sure so far is that with PCR, we can study mutations, multiply target genes to make artificial sequences, and learn about the gene itself.